Work at Animal Biosafety Level 1 is suitable for work with animals infected with biological agents that are characterized and are unlikely to cause disease in healthy workers or other animals. Agents that fall into this category are often referred to as Risk Group 1 pathogens. The animal facilities housing Animal Biosafety Level 1 work are designed to facilitate the cleaning of the room, as well as keeping these spaces reasonably isolated from adjacent spaces. Interior surfaces of animal rooms are water-resistant and easily cleaned and sanitized. The traps of any floor drains are regularly filled with disinfectant. Windows are non-existent or, if present, sealed. A sink for hand washing and cage washing equipment is readily available. The clothing and equipment worn when working in an animal room will be determined by your institution's policies and specified during the protocol's risk assessment. When working in an animal room, a gown or lab coat should be donned over your street clothes or standard uniform. Gloves must also be worn. When donning a glove, check for holes or other breaks and discard any glove that contains breaks. Many institutions also require basic eye protection, such as these safety glasses. This is the minimum level of protective clothing and equipment worn when working at Animal Biosafety Level 1. However, additional clothing and equipment may be specified during the risk assessment to protect animal handlers or immunocompromised animals. Wear a gown, gloves, hair cover, and surgical mask to protect immunocompromised animals from the particles we shed. Replace the surgical mask with a respirator to limit your exposure to animal allergens if warranted. To provide barrier protection from splashes to the eyes, mouth, or face during some procedures, such as this necropsy, wear a surgical mask and a face shield. Again, the exact clothing and equipment worn will be specified for your protocol during the risk assessment. This clothing and equipment may be donned in a locker room, at the entrance to the animal room, or sometimes even in the animal room. Access to an Animal Biosafety Level 1 facility is restricted, and individuals entering must be trained in the hazards associated with the care and handling of animals. At many institutions, they may also receive a medical clearance before working with animals. The doors leading to these animal rooms, generally open inward, are self-closing and must remain closed when experimental animals are present. As with all research laboratories, no human food or drink should be brought into the animal room, and eating and drinking is strictly forbidden. After entering the animal room, the first task is usually cleaning and sanitizing the work surfaces you intend to use to handle animals. At this level of containment, animals have traditionally been housed in cages with wire tops and procedures conducted on open benches. However, to control the spread of aerosols, and particularly allergens, many institutions are housing animals in filtered caging systems, such as this micro-isolator cage, or ventilated cage racks. Special containment devices and local exhaust ventilation may also be used to minimize the release of dusts, and particularly allergens, when opening cages and handling animals or when changing cages. These devices may include a biological safety cabinet, a negative pressure animal transfer station, and a locally exhausted downdraft cage dumping station. Animals at this containment level are generally restrained by hand. Animals are typically moved to the top of the cage where they are more readily grasped. To reduce the probability of being bitten or scratched, animal handlers must be trained in the specific handling and restraint procedures required to conduct their work. These are species specific. New animals should be given several days to acclimate or become familiar with new surroundings before being handled. An acclimated animal is generally less stressed or agitated and therefore more easily handled. When handling rats, consider wearing Kevlar gloves or another cut-resistant glove under your exam gloves. Kevlar gloves can prevent some bites and scratches from breaking the skin barrier. Sedatives may also be used to minimize the anxiety and reactivity of the animal, decreasing the potential for bites, scratches, and needle sticks. If bitten or scratched by an animal, immediately secure the animal and then remove your gloves or other protective clothing and wash the site thoroughly for 15 minutes with soap and water. If your face or eyes become contaminated, remove your gloves and flush your face or eyes for 15 minutes using an eye wash. After cleaning and applying first aid following a bite, scratch, puncture, or splash to the eyes or face, seek additional medical attention immediately. Sharps are devices that can cut or puncture the skin. A sharp may be single-use, such as this needle and syringe, or reusable, such as this pair of scissors and forceps. The use of sharps is not restricted in an animal biosafety level 1 facility. However, it is good practice to try and substitute sharps with non-sharp items, or safer sharps, when possible. When a sharp must be used, the following guidelines will help prevent a skin puncture or cut. Keep the sharps disposal container within arm's reach and dispose of sharps immediately after use. Change out sharps containers when they are 2/3 full and never allow them to overflow. When using a sharp, keep the non-dominant hand out of the cutting area by using forceps or a hemostat. When removing a used scalpel blade or handling a similar sharp, use a hemostat, forceps, or other device rather than your hand. Better yet, use a disposable scalpel that does not require the blade to be removed or cleaned. To clean reusable sharps, place them in a container of disinfectant immediately after use, or spray them with disinfectant. Use a brush to remove any residual debris after the sharp has been sanitized. After removing the debris, return the sharps to the disinfectant and allow them to soak again. All work surfaces, tools, and equipment that become contaminated during your procedures should be immediately decontaminated. Work surfaces should also be cleaned and sanitized after you have completed all your work and are ready to leave the animal room. The wastes generated are collected and removed in leak-proof containers, such as these sealed bags, and may be disposed of as non-biohazardous waste. This assumes there are no chemical or radiological hazards associated with this waste. Animal carcasses are handled according to local regulations, but are typically separated from other wastes, placed in cold storage and later incinerated. If you will be transporting an animal out of the animal room, it must be placed in a secure cage and a cloth or paper drape wrapped around the cage. The cage serves as primary containment and should be transported to and from the animal room in a secure manner. The exact procedure for removing your protective clothing and equipment when exiting the animal room or facility will depend on the configuration of the facility, your institution's policies, and the equipment and clothing worn. In all cases, it is removed in a manner that minimizes the spread of allergens. At this institution, equipment and clothing is removed in a locker room. The gown or lab coat is removed first and placed in the appropriate laundry bin. Next, the gloves are removed. all disposable items are placed in the non-regulated waste stream. Finally, any reusable or disposable eye, hair, face, or mouth protection is removed. After removing your protective clothing and equipment, wash your hands thoroughly before exiting the facility.